Hi, and welcome to Addgene webinar. My name is Benoît Jiquel, and I'm a scientific outreach manager at Addgene. And today I would like to discuss with you uh, about how Addgene can help scientists that work on COVID-19. So at the end of this presentation, you will have the opportunity to ask me questions about the subject. But afterwards, if you would like to reach out to me for other questions, you can get in touch with me at benjikel at gene.org or via my Twitter account at benjikel. So first, I would like to give you some definitions about COVID-19 and SARS-CoV-2. So COVID-19 stands for coronavirus disease 2019. So it's not the actual virus, it's the disease that is caused by a coronavirus called SARS-CoV-2. You can find it as well named 2019 nCoV. It was the first name given to this virus, but now the official name is SARS-CoV-2. And the scientists came up with this official name because it has been shown that this virus is 96% identical to SARS-CoV-1, and it triggers the same syndrome, which is the severe acute respiratory syndrome. So that's why we came with this name that avoid any confusion. So this virus is part of a family of coronaviruses, and these viruses are found in different animal hosts and as well in human. You have this, for example, these four viruses that are in the red square here on this figure that are coronavirus that infect human and trigger common cold. So these viruses are very common, and I'm pretty sure that you have already been infected by these viruses. But from this animal host, and especially from the bat, it emerged some other viruses that can trigger more severe disease. And in 2002, for example, in China, the SARS-CoV-1 emerged from bat and spilled over to humans via civet and small carnivores, and it triggered this epidemic in 2002. In 2012, you have this MERS-CoV virus that emerged as well from bats in the Middle East and via camels spill over to humans and trigger a very severe disease in the Middle East that is still there. And recently in China, there is another virus, the SARS-CoV-2, that emerged as well from the bat and spill over to humans. And in that case, we still don't know if it spill over via an intermediate host. And the hypothesis, for example, is that the pangolin can be this host, or if it goes straight away from the bat to the humans after humans consume bat as food. And after that, you are well known that this virus is very contagious, and for one patient that has the virus, this person can contaminate three other person, and this virus spread out into the world and created this COVID-19 pandemic. So why we call them coronavirus? So if you look at this picture on the left-hand side, you can see this is an electronic microscopy image of the actual of an actual coronavirus. And as you see, it looks like that you have this kind of crown around the virus. And so these two scientists, June Almeida and David Tyrrell in the 60s, came up with this name of this virus that have this crown or this corona. As you can see on the figure on the right-hand side, that this virus, which is an RNA virus, that it contains RNA genome, has an envelope, the smaller envelope proteins, and then in this envelope you have as well a membrane protein, which is a glycoproteins, and also the nucleocapsid protein that bind the RNA genome, and also the spike protein that is very useful for the entry of the virus. But this biology, I will explain it a little bit later. So here's the coronavirus genome, a coronavirus genome. It's long of 30 kb. It's an RNA virus, and this genome um, can be at the origin of 29 proteins. So you have the most obvious, the structural proteins that I just talked about. So the spike protein, the E protein, which is the envelope protein, the N protein, which is the membrane, and also the N protein, which is the ribonucleoprotein complex that will bind to the RNA genome. And you have also other proteins that are non-structural proteins on NSPs, and also accessory proteins. So we don't know all the function of these proteins, but those proteins are involved in the viral replication, the assembly of the virus, and also interfere with host defense within the cells. So how the virus infects the host cells and how it replicates. So this coronavirus needs to interact via the spike protein with the receptor at the cell membrane to be able to end up in the endosome, and then within the endosome, with the help of the target, the host cells protease, the membrane of the virus will fuse with the cell membrane, and the viral RNA will be released into the cytoplasm. There, the RNA, the genome RNA will be replicated, and the gene of the virus will be transcribed and translated into proteins. And those proteins then will be able to reassemble a new virus that will be then this mature variant will be then released into the outside the cells, and where they can infect other cells. So this is a replication cycle for a coronavirus. But what do we know in details about the SARS-CoV-2? Actually, the SARS-CoV-2 is doing the same as other coronavirus, as you can see on the figure on the left-hand side. It's just that it uses some specific receptor or proteins to help that virus to go into the cells. So, for example, the spike proteins of the SARS-CoV-2 interact with the angiotensin converting enzyme receptor, which is ACE2, and thanks to ACE2, this coronavirus enter into the cells. And as you can see on the right hand side, here is the structure of this interaction between human ACE2 and one domain of spikes, which is called the RBM domain, which is interacting with this receptor, enabling the virus to enter. But then it is not well known which protease can be involved in the fusion of the virus membrane and the cell membrane. TPRSS2 is one candidate, and people are working on this protein to see if it's the only one that helps the virus to enter into, or to have its membrane fused to the cell's membrane, of if other proteases like cathepsins can also play a role. So this is a bit of an introduction of what is a coronavirus and what is SARS-CoV-2. Now I would like to discuss with you how plasmid could be useful for research on this virus. And basically there is 4 points that I would like to show you today. And so the first one is that plasmids are very useful to understand the basic biology, because as I said earlier, we know some facts about coronavirus, we know some facts about SARS-CoV-2, but we still don't know everything about this biology, and there's a lot of things to discover yet. Also, the plasmid can be used for diagnostics, and some new techniques actually are really good to do this in your lab. And plasmid as well are very useful to develop therapies and also to develop vaccines. So if we go back to the biology of SARS-CoV-2, as I said earlier, we know that receptor ACE2 is the receptor for spikes, but either are there other receptors? It's still a question mark, and we know, for example, that on this spike protein there is a site for cleavage that is normally triggered by a protease named furin. So, is the furin trigger this cleavage, and is it helpful for the virus to enter into the cells? Is still a question mark, and a plasmid expressing furin and ACE2 will be very useful to answer this kind of questions. For example, as well, we know that some metalloprotease can cut the receptor ACE2, and in that case, the virus cannot enter, and we still don't know why it is happening, and so a lot of people are working on that as well. As I say as well, what are the exact role of TMPRSS2? Is it to fuse the membrane to the cell membrane? Is the cathepsin are involved as well in this process? Are there other proteases that play this role? So it's still some question mark here. And as I told you as well earlier, there is a lot of gene into this virus that can impair pathways within the host cells and decrease the host defense. And here as well, even if we know some role that play those viral proteins, a lot of them we don't know at all what they are doing. So, for example, the NSP2, which is not clear what this protein can do, can bind to protein involved in intracellular trafficking, but what exactly the protein is doing, we don't know. NSP11, we have no clue of whatsoever or what this protein is doing, and the NSP13 might help in RNA unwind, but it's something that has to be discovered as well. And the other protein, even if we kind of know their function, we don't know all in details. And having those proteins expressed in plasmid could be very useful to understand their functions. So what do we have here at Addgene to help the scientists try to answer this question? So we have a lot of plasmids, about 900, more than 900 plasmids that are coming from 23 labs, coming from everywhere in the world. And this plasmid can express H2, for example, TMPRSS2, cathepsin L. We have plasmid that express ADAM17. They have plasmid that express furin as well. And on the other side, so on the virus side, we have plasmid that express all the proteins from the SARS-CoV-2. So the mature non-structural protein, the accessory proteins, and structural proteins as well. And also we have a partnership with the biotech company Gingko Bioworks, and they want to have to create a collection of expression plasmid for this COVID-19 research. And the idea is to really synthesize different gene of the virus and to put them into expression plasmid and attach those genes as well with different tags, and with the idea that it would be useful for the research community. So they already have created a lot of this plasmid, and the idea is to create around 3,000 plasmids. And so right now we have at Addgene bacterial expression. So we have SARS-CoV-2 gene that are can be expressed in E. coli with different tags. So you can then use this plasmid to express the protein within cells or for purification. We also have a system where you can express those genes into yeast. And it's now available at Addgene. Recently, we have as well mammalian expression for this SARS-CoV-2 gene. That's exactly the same as bacterial and yeast expression system. You have all these genes for SARS-CoV-2 that are fused with tags or other proteins for protein expression or purification. We also have a SARS-CoV-2 spike gene construct that is truncated, so enabling people to make like VSV pseudovirus, and also for controls and also to continue to work on those coronavirus. We have as well the common human coronavirus nucleocapsid gene that are in mammalian expression vectors. If you're interested into this collection, you can just go on our website, and there is a blog as well that explains the collection. So as I said earlier, plasmid also can be used for diagnostic. So as I say, SARS-CoV-2 is an RNA virus, and to detect this virus, we just have to extract the RNA from these viruses that are in the swab, and then you do a reverse transcriptase to have the cDNA of this virus, and then you do a real-time PCR to see the viral load that you have in your patient, for example. And with plasmid now, you have this, for example, RTX, which is a thermostable reverse transcriptase, which is as well a DNA polymerase. So basically, it's in one enzyme that can do the reverse transcriptase and can do as well the PCR. So you can use that directly in your lab, and you can test the people that have been infected by SARS-CoV-2. Also, you have some CRISPR-based tool that can be used quite easily, actually, to detect the virus. So it can be used with the Cas9, so the normal Cas9 that will using a guide RNA that is specific for viral DNA. And this Cas9 will cut this viral DNA, and then on an agarose gel, you can see if there is a cleavage or not. Also, there is other tests that involved Cas12a, which is called detector, or Cas13a, which is called Sherlock. And in that case, it would be a collateral cleavage. So basically, you put this two enzyme with guide specific for the viral RNA or the viral DNA for the Cas12, and then once they find it, they will start to cut this RNA or this DNA, but also they will have a trans effect on repertoire molecules, and they will cut the repertoire molecules that have, for the Cas12 and for the detector, this repertoire molecule has a fluorescent protein on one side and a biotin on the other side. And the fluorescent protein is quenched at that stage, but if there is a cleavage, the biotin will stay on the control line with the strip, because on this flow strip you have a streptavidin, and then the part with the fluorescent protein will move with the flow strip and will be captured by an antibody. So if you have a cleavage, you will have this second band that is due to the antibiotic capture of this cleaved reporter. And if you don't have any virus, for example, you don't have this cut, and you don't see this second line. And for the Cas13, it's the same principle in the sense that the fluorescence is quenched, and if you cleave the RNA, the reporter molecules, then you have the fluorescence that is triggered, and you have the visualization of this fluorescence. If you're interested in these techniques for diagnostics, we have a couple of blogs that can be very useful, and also we have a protocol where you can find a kit-free RNA extraction. Plasmid can be very useful as well to develop therapies for COVID-19. So I just put the figure of the replication of the SARS-CoV-2. And it's obvious that some drug that we already use can be repurposed to treat people with the SARS-CoV-2. So people came, for example, started to test the chloroquine or the hydroxychloroquine uh, to see if it can help uh, with the entry or impair the entry of the virus. And I don't know if you saw the latest news, but it has been shown that this doesn't do anything on the disease at all. And also some drugs regarding replication or the RNA replication can be used, like the remdesivir or the favipiravir. So right now, those drugs are giving some hope, but still we don't know if can be used to treat a patient. Also, there is these two drugs, the camostat and nafamostat, that can trigger the TMPRSS2. Right now, some people are testing those drugs, but plasmid can be useful when it comes uh, to avoid the entry of the plasmid via the ACE2 receptor. Because in that case, for example, so normally, as you can see, the SARS-CoV-2 infection goes via the ACE2 receptor. But if you can come with an RBD domain that you can just express this soluble RBD domain of the spike. This RBD domain is the domain that will interact with the ACE2 receptor. In that case, you will then impair the interaction between the virus and the ACE2, and then you will impair the entry of the virus. Similarly, you can also express an anti-ACE2 antibodies that will be blocking the virus entry, and so the virus cannot enter to the cells. Also, using plasmid, you can engineer neutralizing bodies. So in that case, you use the ACE2 the ACE2 receptor that you fuse with the Fc of an antibody, and this ACE2 Fc, for example, will be used as a decoy and will interact with the virus. And thanks to the Fc receptor, immune cells can then get the virus and destroy the virus. Other people were thinking of trying to find proteins within the host cells that will interact with the virus that we don't know yet, and to see if those protein has already drugs. So some people, they just take the whole virus, all the gene of the virus, they put them into expression vector, and then they express those proteins in cells, and then via spectrometry they just look at the interactome of this protein with the host, and then from that they found out new targets for therapies that can be used that use approved drugs or experimental drugs. So they came up with a list of drugs that would be potentially useful therapies against the COVID-19. Also, plasmid could be useful to develop vaccines. So usually vaccines are, we take a live attenuated virus that we inject into the people, and then this virus will trigger the production of antibodies against this virus, and the people would be vaccinated against this virus. What can be done as well is that we can express or produce a viral vaccine. So just by having just one part, like the surface protein of the virus that is expressed into another virus, And this virus, we just have to put this virus into a person, and then it will trigger as well antibodies against this specific protein that is expressed in this virus. You can also have DNA vaccines. In that case, it's very simple. You take the plasmid and you inject into the people, and you have as well antibodies that are produced again. That's the protein that is expressed by this plasmid. You can also express directly RNA. So from the plasmid in vitro, you express RNA, and then you do RNA vaccine. And it's the same principle. And also you can express directly the protein and inject the protein. And you have this kind of protein-based vaccine that at the end, like the other methods, will help with the production of antibodies against the protein that is on the surface of the vaccine, of the virus, sorry. So now I would like to go into our website with this video where I will show you what is exactly how looks like our resources page, and how you can find the plasmid that will be useful for your research. So basically, you can go on Addgene.org collection COVID-19 resource, and you will arrive on this page where you will have a small background about the pandemic and the virus. You will have as well links to the resource that are at Addgene's, which is protocol, research articles, and other plasmids. Then you have some plasmid that are highlights. And then you can see that we have a paragraph about the Gingko collection. And we have actually a special collection page for this collection that I'm going there now. So you can see that in this page you have a background of this project, and then all the list of all the plasmid that you can order to get access via our website. Also, you have as well the plasmid that are first for the virus. So the virus plasmid, then the virus for SARS-CoV that can be used as a control, and then the plasmid that express human proteins or target proteins. Then you have this Sherlock and detector tools that can be used for diagnostics. And also you have external resources, protocols, and also access to other resources. So as I said earlier, right now in our collection, we have more than 900 plasmids coming from 23 labs. And we are still getting more and more plasmid because the community is really want to share with the rest of the community, and also want to share with the industry. And we have actually as well put in place an MTA that allows some of the plasmid that can be ordered by pharmaceutical industry, for example. And so if you're interested in those resources that are changing, you can go on or subscribe to our Addgene newsletter. We'll give you some news there. Follow our Twitter feed, where we will give you a lot of information about these resources. And of course, our Addgene website, where you can find news about these plasmids. And if yourself you are working on the COVID-19 and you are producing some interesting plasmid, you can deposit them at Addgene, and I will be happy to help you. So feel free to contact me if you're interested, or also you can contact our team at deposit@addgene.org. The process is quite simple, because what we just need is all the information of your plasmid, and then a couple of microliters of your DNA samples. And once those plasmids will be at Addgene, we will store them, and we will do some quality control on them, and we will basically sequence the full plasmid. And then the deposit will be complete. Your plasmid will be available online, and for yourself it will be interesting, because you can increase the visibility of your research, and the community can have access to really great tools. So to finish it up this webinar, I would like to thank all my colleagues at Addgene that are working very hard to, especially those days, to have this collection, especially for COVID-19, that is available for the community. So a great thanks to them. And I also wanted to thank all the labs that have deposited the plasmid to Addgene.